Okay, so thank you, Alain, for the invitation. It's always a pleasure and an honor to speak in this venerable house. It's also very nice to be in Paris in May. So what I want to discuss today are two stories that relate to slightly different ways whereby morphogen gradients are used in the embryo, maybe slightly more dynamic. And they have to do with what I listed here as what I call cardinal features of the vertebrate body plan. One is the elongation, the fact that in vertebrates the body axis is an elongated structure. Second one, which I'll discuss, is the segmentation, namely the fact that it's composed largely by repeated anatomical modules, like the vertebrae here or the somites in the embryo. And the third one is the bilateral symmetry. And the last one would be the regionalization, namely the fact that you have a head and a tail and so on. So today I want to essentially focus both on segmentation and bilateral symmetry and try to tell you how gradients are used in these two processes to set up these features of the embryo. So first I'll start with just by introducing segmentation, and so just want to remind you that in invertebrates, segmentation arises from the formation of the embryonic somites, which are epithelial structures that are generated in a rhythmic fashion at the tip of a tissue, which is called the presomatic mesoderm, which itself derives from gastrulation at the level of the primitive streak and the tail bud. And so this tissue here forms the so-called paraxial mesoderm. And one of the very striking characteristics of the segmentation process in invertebrates is the fact that it's a rhythmic process. Namely, somites are added with a defined rhythm, which is specific of the species. So for instance, in chicken embryo, as you can see on this movie, the somites are added with a 90 minutes periodicity. And, but in other species, like mouse, it would be 2 hours. In zebrafish, it's 30 minutes. In human, it's not known. And so we've been interested for many years in trying to understand the molecular control of this rhythm. And now we know that this rhythm is controlled by a molecular oscillator that we term the segmentation clock, which corresponds to a dynamic set of properties which result in the production of periodic waves that propagate rhythmically along the presomatic mesoderm, that is the precursors of the segment, with a period that corresponds to the time to produce a somite. And so the idea is that these periodic waves of the segmentation clock actually control the rhythm that's involved in segmentation. So we understand now some of the circuitry that underlies these molecular oscillations. We know that they involve the dynamic regulation of 3 major signaling pathways, the FGF, the Notch, and the Wnt pathway. And these 3 pathways are activated throughout the presomatic mesoderm, the tissue that experiences the oscillations. And that leads to the cyclic activation of these genes here that are figured in red or in blue, and which largely correspond to negative feedback inhibitors of these pathways. And so the idea that underlies most molecular oscillator models that have been proposed to explain this periodic behavior argue that these oscillations are triggered thanks to negative feedback with delay system. And so there the idea would be that you have constant activation of the system, triggers the activation of the negative feedback inhibitors that will shut down the activation of the system, and by the same token shut down their own production. And so at some point they will get degraded, and when they get degraded, the activation resumes. So most models have been centered on this cyclic gene here called the HES or HES7 in the mouse, or the HER genes in fish, which are negative, which are transcriptional repressors that can negatively regulate their own expression. And so but I don't think they really reflect the whole complexity of the system, but that's just to give you an idea of the general principle. So we've been also able to engineer a real-time reporter to visualize these oscillations. And so, for instance, this is work from my lab from Alexander Olela when he was a postdoc in the lab, when he engineered such a reporter by fusing the promoter of one of the cyclic genes, lunatic fringe, to a destabilized fluorescent protein, Venus, and then made the transgenic mouse. And this is what you see here. So this is this reporter mouse lying on the side. This is the left side. You can see the somites here. And this is the presomatic mesoderm that experienced these traveling waves of lunatic fringe expression. So what you can note here on this movie is one that what you're visualizing is the expression of a notch target. So you're looking at the dynamic behavior of notch, and you can see how tightly coordinated this is. And second, what you should note also is that this process terminates by the production of a stripe. And so this stripe is thought to be the pre-pattern, the genetic pre-pattern on which the future segment is going to be formed. So the clock is thought to be the system that controls the rhythm, the temporal aspect of segmentation, but you need a second component in order to produce a periodic series, a spatial periodic series. And so this second system is thought to involve a set of gradients, of posterior gradients of FGF and Wnt signaling. So this is, for instance, the expression of FGF8, one of the FGF ligands, the mRNA level, which forms a posterior to anterior gradient, which is antagonized by a system of an anterior gradient of retinoic acid signaling, which is produced by so this is the RDH2, which is the biosynthetic enzyme for retinoic acid, which is expressed in the somatic region. So these two sets of gradients antagonize, and this is thought to play a role in the positioning of the segmental response, that is the formation of the genetic stripes that will ultimately become the segment. And so more dynamically, the way we view the system is based on these two components, namely the clock and the system of gradient, which is often referred to as the wavefront in reference to a classical theoretical model put forward by Jonathan Cook and Chris Zeman in the 70s, where they proposed that the segmentation could be explained by the displacement of an oscillator in space, which is pretty much what we have here. And so what we have is the clock, which delivers these traveling waves of signaling across the entire presomatic mesoderm, but only the cells that pass the determination front or the threshold, which is defined by this system of gradients, only these cells become competent to respond to the clock signal. And when they get hit by the clock signal, as you see here, then they activate the expression of specific genes, such as the gene, for instance, MES2 that you see here. So we've been interested in the molecular circuitry that underlies these oscillations for many years. And most of what I've shown you so far was generated in the embryo, and that's, I think we reached pretty much what we could achieve using classical techniques such as for genetics and so on. And so we've been trying for some time to design an in vitro system that we could use in order to study the oscillations. And so the first breakthrough in the area came from work from Alexander Olela, who was now his lab at EMBL. And what Alex did is that he designed a system where he showed that if you take the tail bud from the reporter mouse that I showed you that he had generated in my lab, if you take this tail bud, put it in culture, it will form a disk of cells which continue oscillating. So this was really showing that you can maintain this oscillatory behavior in vitro. And, but there was something which we were not completely happy with, is the fact that Alex's system essentially recapitulated development in the sense that the cells would oscillate for a bit and then they would stop and make a segment and differentiate. What we wanted is to generate a system where we could maintain the cells oscillating indefinitely in order to be able to dissect properly the mechanism of the oscillations. And so this was work from Alexis Hubo in my lab, a PhD student, who used the system initially developed by Alex, but modified it using protocols that were established in the lab by Jérôme Chal to differentiate embryonic stem cells to a presemitic mesoderm fate. And when he did so, when he combined the two systems, what he could obtain are explants of this transgenic mouse in which the oscillations were maintained stable for a very long period of time, for at least 48 hours. That is more than 25 oscillations, so a lot more than they would normally do in vivo. And so using this system, Alexis started to dissect the dynamic property of the oscillator. And the first thing he showed is that when you treat these explants that normally oscillate with a drug that blocks notch signaling, then you lose the oscillations. This is not unexpected because the reporter is a notch reporter, so that's so far so good. But in the field, there's been a controversy about the role of notch, people proposing that notch is important for the synchronization of the oscillations, so the generation of these smooth waves along the presemitic mesoderm, and that's mostly work from zebrafish, while people working in mouse were mostly arguing that it was important for the oscillations themselves. So clearly that shows that in the system, notch is important for the oscillations. You can see this quantified here. You see that when you treat with this notch inhibitor, DAPT, then the oscillation gets damped and the rest. So then Alexis used the system to probe a number of concepts that were proposed in the field, and an important one was the assumption that the cells of the presemitic mesoderm harbored an intrinsic oscillator. And so this was quite easy to test in this system, and so what he did was just to dissect the tail buds, to dissociate this to single cells, and plate this at low density on fibronectin. And when he did so, then he imaged the cells, and as you can see here, what happens is that the cells do not maintain the oscillations. They essentially progressively lose the expression of the reporter, and if you look at the tracks, you see that there are no real stable oscillations in these conditions, so they get essentially damped and erased. So you see only bursts of fluorescence, but which do not correspond to a real oscillatory pattern. So, but what Alexis noticed also is that while the cells stop oscillating, this is not because they differentiate, because if you look at the expression of markers that are specific for the oscillatory domain, the posterior presemitic mesoderm, namely this gene, this protein TBX6, you see that the cells that stop oscillating retain an identity of posterior presemitic mesoderm. And to further test this, what he did is to reaggregate cells that had stopped oscillating. So he cultured dissociated cells overnight, and then so they stopped oscillating, and then he reaggregated the cells. And as you see here, the cells that had stopped oscillating, they can resume oscillations once you bring them back to a certain density. And so this experiment was arguing two important things. One is that the cells don't harbor intrinsic oscillators because you can switch it on and off, and so suggesting that these oscillations are rather reflecting a dynamical property of the system. And then second, it was telling us also that one key parameter is the cell density, because once you isolate the cells, so I should emphasize that the explants and the single cells are cultured exactly in the same conditions. And in one case, when the cells are isolated, you lose the oscillations. When they're cultured at high density, you rescue the oscillations. So we were wondering about the signal which is associated to density that controls this, the onset of the oscillations. And, and so this is just the quantification of the tracks. And actually, by doing a negative control, Alexis made quite a striking observation, which is that if you culture now your dissociated cells, not on fibronectin, but on a simple substrate, just a BSA coating, what you see is that the cells now, even the isolated cells, resume normal oscillations. And these oscillations exhibit a period which is the one which is seen in a mouse embryo, which is about 2 hours or so. So this was a bit puzzling because the same cells, when cultured on fibronectin, they would not oscillate, whereas on this poor substrate, they were starting to oscillate. And so, in fact, what we realized is that the big difference between the cells cultured on fibronectin and on BSA is their adhesive behavior, meaning that the cells on fibronectin are very spread out on the substrate, whereas on BSA they're very confined, so very different organization of the cortical skeleton. And this is known to be associated to the activation of a mechanosensing pathway, which is well known, which is the YAP signaling pathway. So in the cells cultured on fibronectin, YAP is known to be nuclear. And when you looked at that, indeed, on the isolated cells on fibronectin, it is nuclear, whereas on BSA, YAP is not enriched in the nuclear as on the fibronectin. So I'm not showing you all this data because this was recently published, but that was suggesting that YAP signaling could be an important switch in the control of the oscillator. And so Alexei was able to show that by now treating cells cultured on fibronectin that would stop oscillating with a drug that inhibits YAP signaling, which is latranculine A, which arguably does a lot of other things than inhibiting YAP signaling because it blocks the polymerization of actin. But surprisingly, when you treat the cells that normally stop oscillating with this drug, what you see is that they resume normal oscillations, even though they're very clearly strongly affected by the treatment. And so that suggests somehow that YAP signaling, a high level of YAP signaling oscillation is able to block the oscillations. And in fact, the way you could ensure that this behavior is linked to YAP signaling is by infecting these cells with an antivirus that expresses a dominant activated form of YAP. And that completely blocks the oscillations in this context. So one last observation I should mention, I won't show you the data, but it's important to mention it. It's that surprisingly, this oscillatory behavior, which is rescued by latranculine A, which is associated to, which is visualized by the oscillations of this notch reporter, cannot be suppressed by the treatment with the notch inhibitor, the DAPT, that blocks the oscillations in the explants. So how do we make sense of these experiments? Then we teamed up with Mahadevan, who's a physicist in the Department of Physics at Harvard, who suggested that one way to think about the system was to consider that it behaves as an excitable system. So we've heard a bit already about excitable systems. The most classical one would be like the neural action potentials, but you can think also about the heart calcium waves or the Dicty CMP oscillations. They're characterized by the fact that they can exist in a quiescent state, and upon stimulation, with a small stimulation, they will generate a small excursion. This is an analogy of a pendulum here. They will just make a small excursion and then come back to their equilibrium position. But if you produce a strong stimulation, which is above a certain threshold called the excitability threshold, then the system will make a large excursion, and this excursion is characterized by being refractory to further signaling, and then it will come back to its position. And if the stimulus is sustained, then it will trigger oscillations. And so that's the way we interpret this data, is just by postulating that a notch in this context acts as a component of the stimulus, a bit like the current in the action potentials, and is able to trigger the system above the excitability threshold, which is controlled by YAP signaling, and that leads to the dynamic response, that is the oscillation that we see. And thus, when the cells are isolated and cultured on fibronectin, then YAP is activated, as I showed you, which means that you essentially raise the excitability threshold, and so your notch stimulation is not enough to move the system, and so it failed responding and it's not oscillating. And now, in contrast, if you block YAP signaling with the latranculine or if you culture the cells on BSA, YAP is low, so your excitability threshold is very low, and then your system becomes hyper-excitable. And so then you see spontaneous oscillations of the system. So this was the conclusion of these experiments. It was, for us, an important conceptual change compared to the way we were thinking about the segmentation clock, which was essentially thought in terms of ad hoc oscillators that were coordinated together through notch signaling, and that now offers us a conceptual framework to really think about how these oscillations are generated. But the system that I described was not completely satisfactory because you need to microdissect embryonic tail buds, and so you cannot have access to many cells. And so for a long time, we've been trying to also develop in vitro systems based on human ES or iPS cells, or mouse and human ES and iPS cells, differentiated to the presymmetric mesoderm with the hope that we could be able to generate oscillations in this system in vitro. That would be first provide us with great in vitro system to perform molecular experiments, but also would tell us about the embryology of the human segmentation, on which nothing is known, because this takes place between 20 and 30 days, so way too early in the embryo, 3 and 4 weeks, so way too early in the human embryo, so there's essentially nothing known about this. So anyhow, so we had shown that if we take pluripotent stem cells, such as embryonic stem cells or iPS cells, we treat them with a Wnt activator and a BMP inhibitor, they show characteristics of these neuromesodermal precursor cells and eventually become posterior presymmetric mesoderm. And so the way we monitor the differentiation to the posterior presymmetric mesoderm fate was by engineering reporter lines, where fluorescent reporters would be placed under the control of promoter of genes such as mesogenin-1, which is this gene here expressed in the oscillatory domain of the presymmetric mesoderm. And so when you look at what happens in cells that are treated in this way, you see that at day 1, you don't see much happening yet, but at day 2, we see massive activation of this posterior presymmetric mesoderm reporter, which is maintained for another 2 days. And if we quantify this, we see that this differentiation is extremely efficient with more than 90% of the cells that turn up the posterior presymmetric mesoderm markers. So Margarete Diaz-Quadros, who's a PhD student in the lab, started to study this in detail. And so she looked at the expression of a number of markers during this differentiation sequence. And so, for instance, here she looked at the expression of SOX2, which is a marker of pluripotent cells, but also of neural cells, and brachyury, which is a marker for mesodermal cells. So you see that initially at day 0, all the cells express the pluripotent marker SOX2. At day 1, they maintain SOX2, but they start to activate brachyury, which means that they acquire a state that resemble these NMPs, which has been also nicely studied by James Briscoe here. And then these cells turn off SOX2, and they maintain brachyury for some time, and then eventually they lose brachyury. And in parallel, they will activate TBX6, which is this marker of the posterior presymmetric mesoderm, indicating that the cells, compared to the embryo, they follow a very similar differentiation sequence, starting with so-called NMPs, then MPCs that would correspond to this stage where they still have brachyury and TBX6, and then the posterior presymmetric mesoderm with TBX6. What is very striking in this system is that not only that it very accurately recapitulates the embryonic sequence, but also at the cellular level. And so if you look at the colonies, for instance, at this stage, cells are essentially epithelial, like in the epiblast. And then between day 1 and day 2, they massively become mesenchymal, which means that they undergo an epithelium to mesenchyme conversion, which is very much like gastrulation in the embryo. And so then Margarete started to look at the the signaling, so this FGF and the Wnt gradient in the differentiating cells. And so as a readout, she looked at DP-ERK, which marks the activation of ERK, which is downstream of FGF signaling in this context, and the beta-catenin, which is, when it's nuclearly localized, reflects Wnt activation. And as you see here, at day 1, there is activation of both, there's expression of DP-ERK and nuclear beta-catenin, meaning that there is activation of both pathways. Similar at day 2, which means that we're in this equivalent region of the high level of the gradient. And then at day 3, there is a sharp decrease of the signaling by DP-ERK and beta-catenin. So again, the differentiation sequence nicely recapitulates the developmental stages of this lineage as we understand them. Now, what is a bit striking that you might have noticed is if you look at what is added in the medium during these days. So for day 1 and day 2, we had KIR, which is a Wnt activator, and LDN, which is the BMP inhibitor. But there is no FGF at these stages, which is added. Yet there is strong FGF activation that takes place. In fact, FGF signaling is shut down when FGF is added in the medium. And we can show that this signal is specific to FGF because it can be blocked with an inhibitor for the FGF receptor. And if we look now at Wnt signaling, here, chiron is a Wnt activator, and it's present throughout the culture. Yet we see a strong downregulation of the nuclear beta-catenin that takes place at day 3, exactly when it's expected, despite the presence of chiron. So what I'm trying to say is that these cultures, in fact, do not behave as a white page, as I guess I initially thought they would, and that the important thing was to essentially recapitulate as closely as possible the developmental sequence of growth factor, which is experienced by these cells during development. In fact, what this tells us is that they show a very high degree of autonomy and self-organization, a bit like the organoids. So now if we move 1 day later, what we see is that at day 3, the cells express the homologue of this gene, MES2, which means that they've reached the so-called determination front and activated the segmentation program. So the next question, so together that really suggested that these cells differentiate exactly as, for instance, mouse presemitic mesoderm cells or chicken presemitic mesoderm. And to further characterize these cells at the single cell level, what we did is to perform single cell sequencing of these cultures at day 0, day 1, day 2, day 3, and day 4. So we sequenced almost 15,000 differentiating human iPS cells, and with the help of Daniel Wagner from Alan Klein's lab. So we used the InDrops platform for that. And there are 2 interesting things that came out of this analysis. One is that at the gene expression level, this recapitulates exactly what I was showing you with the antibodies and so on. That is, the cells become first NMP-like cells, then they activate posterior presemitic mesoderm marker, then they reach the determination front, and then the anterior presemitic mesoderm. And so that's one thing. But the most striking thing is that what you see here is an unbiased clustering of all the cells, which has been done. And in fact, the cells cluster spontaneously in this unbiased approach. They cluster spontaneously by day, which means that the cultures are extremely homogeneous. All the cells reach the same stage at the same time. There's no heterogeneity in these cultures, which to me was a big surprise. But which means it's a great experimental tool, because what you have here are very, very synchronized cell population with respect to their differentiation. And that will greatly simplify the analysis of molecular processes that happen during this developmental sequence. And so, of course, what we wanted to look at is the oscillations in this context, because everything seems to be right, so we would predict that they should oscillate. And so what Margaret did was to introduce, using CRISPR-Cas9, a destabilized fluorescent protein called Achilles, generated by Miyawaki and Kageyama. And so she introduced this after the exon 4 of the cyclic gene S7. And lo and behold, these cells, when you look at them under fluorescence, they nicely oscillate at day 2, which is the time when they're supposed to be in the oscillatory state. So this was quite interesting and revealed a predicted period for the human segmentation clock of 5 hours. So this fits nicely with what we know about the difference between the developmental timing of mouse and human embryos, because human embryos are known to develop about 2.5 times slower than the mouse embryo. And the mouse embryo is 2 hours. So then she started to use the system to address a few questions that were difficult to tackle in the embryo. And one of the questions that she addressed is about the role of the FGF gradient on the oscillations. So I told you that the main idea was that the FGF gradient serves to establish a threshold at which the cells become competent to respond to the clock and activate these genes like MES2 and stop oscillating. So what she did was to look at the effect of inhibiting FGF signaling on the oscillations. So here she used 2 different kinds of inhibitors, and what you see is that both of them essentially progressively shut down the oscillations, so they lower the intensity level, but they also progressively arrest the oscillations. But what we didn't see is an immediate arrest. So the arrest is delayed, which tells you that it's not a simple threshold system. The other thing which was quite striking, which is illustrated here, is the fact that as soon as you treat the cells with the drugs, then they become immediately in opposite phase compared to the controls. So that means that the clock is acting not only on the periods and the control of the oscillations, but also on the control of their phase. And there is this, with both drugs, we see this 180-degree phase shift between the control and the treated embryos. And interestingly, this is the effect was dose-dependent. That means that the higher the dose you apply to the embryo, the less oscillations you see, which means that there is a real quantitative control of FGF signaling on the dynamic properties of the oscillator and not a simple threshold model as we thought. Then she looked also at the effect of notch, and what she saw is that when she treats with this drug DAPT, essentially she loses the oscillations as in the mouse embryo, as I showed you earlier on. And she performed similar experiments as the one I showed you from Alexi on the mouse embryo. So when she dissociates the cells, these oscillating cells, these oscillating human cells, then they lose the oscillations if they're cultured on fibronectin. But if you treat the cells with latranquilline A, you can rescue the oscillations. So that's the same as for the mouse. And if you treat with latranquilline with DAPT, you also rescue the oscillations. There's no effect on inhibiting notch in this context, which is interesting because in this case, this is not the same reporter as the mouse. So that's the second example we have where using a notch reporter in cells that are in which YAP signaling is inhibited, in which you cannot block the oscillation of the notch reporter with the notch inhibitor DAPT, which suggests simply that we don't completely understand how notch signaling is working. So conclusion from this part is that what's, to me at least, is one of the most important observations is that we can trigger an oscillatory transition by playing on the YAP pathway or on the density, suggesting that there is a sort of dynamical quorum sensing important for the control of the oscillations, and that involves notch and YAP signaling. We also see that there's no simple threshold system for the wavefront for FGF signaling, which appears to control dynamical parameters such as period, amplitude, and I should have added phase also of the oscillations. And the human oscillator appears to show very similar dynamical properties to the mouse segmentation clock, which further argues that the system works as an excitable system and also provides us with a great tool to dissect this system in vitro. So now I just want to finish by telling you about another aspect of the development of the somites, which has to do with the bilateral symmetry of the system. So you heard yesterday Julien telling you about the system that breaks the symmetry of the embryo. The embryo, when it's generated, is originally bilaterally symmetrical, very quickly bilaterally symmetrical, and the symmetry is broken to give rise to the internal organs. But there is a set of structures that remain bilaterally symmetrical, namely the ones that I discussed that are derived from the paraxial mesoderm, but also others, such as the lateral plate derivatives. And so while we know a lot about how the symmetry is broken and the cilia system and the nodal pathway and so on, we understand very little about how the symmetry is maintained, is first established and then maintained in the embryo. And I think this is a much more complex problem, in fact, than to understand how this is broken. Because if you think about it, to develop these two sides as mirror images over such long period of time is quite mind-boggling, in fact. And so the first important result in this area was generated by Julien Vermeaux when he was a kid working in Pascal Dollé's lab, where he noticed that ALDH2, which is the biosynthetic enzyme that is involved in the production of retinoic acid signaling, when you knock out this gene in the mouse, what you see is that the embryos exhibit a striking desynchronization of somatogenesis between the left and the right side, such that somatogenesis becomes delayed on the right side. And then Julien came to my lab at the time in Kansas City, and he tried to look at what is responsible for this, what controls this asymmetry of somatogenesis. And what he showed is that this synchronization of the somatogenesis in the ALDH2 mutant is, in fact, in response to the left-right patterning system that we heard about. And he could show that in the chicken embryo, and I won't show you the data, but he showed this also in the mouse by crossing the ALDH2 mutant with an inversus viscer mutant, which is a mutant that shows a defect in the cilia and hence shows randomized situs. And in these mutants, he saw that in a fraction of the mutants, the somatogenesis was reversed. So this is quantified here. Normally, in ALDH2 mutants, all the asymmetric mutants are right-sided, but in the double mutants, now you see an almost equal proportion of right-sided and left-sided. So this was suggesting that what retinoic acid signaling is doing in this context is to buffer the desynchronizing action of the left-right machinery. And then Gonzalo Neto was a PhD student at the time in the lab in the US, started to study another mutant called open mind, because in these mutants, the neural tube doesn't close at the level of the brain. And this, the mutated gene is a protein called atrophin-2, or RIRI, or grunge in the fly. And it's a chromatin remodeling, a large chromatin remodeling with chromatin binding domains like a bromodomain or a SEN domain and so on. And what Gonzalo noticed is that this mutant also has a striking desynchronization defect. And in fact, it's a very good phenocopy of the ALDH2 mutant. What was surprising is that this ALDH2, as I told you, this is the biosynthetic enzyme for retinoic acid, so it's probably the strongest mutant in the retinoic acid pathway, at least at these stages. And this gene had never been associated to retinoic acid signaling. So then what Gonzalo was able to show is that, in fact, this RIRI atrophin-2 is a coactivator for the retinoic acid pathway. And so, for instance, when he uses a reporter for retinoic acid signaling, like this RA-LAGZ mouse, that in which the LAGZ gene is fused to a retinoic acid responsive element, when he crosses this reporter in the background of the open mind mutant, you see a very strong downregulation of LAGZ, suggesting that indeed this gene is important for retinoic acid signaling. So in the open mind mutant, there is also a striking phenotype at the level of the components of the clock and the wavefront. And so what Gonzalo saw is that there is a striking desynchronization of the FGF8 gradient, which also pushes anti-early DRDH to gradient and leads to the desynchronization of the stripes that is visualized in this delay of somatogenesis. So the conclusion of these experiments, and I really don't have time to show you all the arguments, but was that a slight asymmetry of retinoic acid signaling here on the right side, which is figured on the graph as this excess blue label here, is going to is important to maintain the symmetry of the FGF gradient. Because if you remove retinoic acid signaling, what happens is that you see this shift, this anterior shift on the right side of the FGF gradient. And what I won't have time to show you is that this, the conclusion of these experiments, in fact, is that what retinoic acid signaling is buffering against in this context is not a desynchronizing action of nodal, but one of FGF signaling, because nodal is an important left-right determinant, but FGF is also another left-right determinant. And so the conclusion here is that the system is important to maintain the bilateral symmetry by maintaining an antagonism between retinoic acid signaling and FGF signaling. And so in parallel to these experiments, Mike Rebagliati, who was a postdoc in the lab, started to look at this in zebrafish. So what the lab of Juan Carlos Espizua Belmonte had shown that in zebrafish, if you block retinoic acid signaling, you get the same sort of asymmetries of somite formation that I showed you. And so Mike used a drug called BMS, which inhibits retinoic acid signaling, and what he saw are also this same desynchronization. But what he also noticed is that in these embryos, there were also a fraction of the embryos that showed left-right defects of the hindbrain. So these are labeled with CROX20, which marks the rhombomer 3 and 5. And as you see, in some cases, there is half of the rhombomer 5 which is absent, and that can happen on the right or on the left side in this case. So the asymmetry of the zebrafish brain has been well studied at the level of the diencephalon, and it's well known that the habenular system is very asymmetrical in fish and in amphibians. And so in the epithalamus of the fish, there is a nucleus which is completely asymmetrical called the parapineal, which influences the asymmetric development of the lateral habenulas that projects to the dorsal interpeduncular nucleus. And so this system, this asymmetry that's visible through the different morphology and projections of the habenular nuclei and the localization of the parapineal gland, the system is known to be downstream of nodal, the same nodal system that Julien talked about yesterday. And one of the characteristics of this asymmetry is the expression of left T1 on the left side in the diencephalon. So we wondered whether the effect of retinoic acid signaling that Mike had seen in the rhombencephalon was linked to this left-right patterning system of the diencephalon. And what Mike did was to look at the expression of lefty in embryos that were treated with this inhibitor of retinoic acid signaling. And as you see here we have only a right R5, here we have only a left R5, but in all cases, lefty was found to be on the left side, suggesting that these asymmetries generated by retinoic acid inhibition in the hindbrain are independent from the system that patterns the diencephalon. So what do we know about asymmetries in the brain? We know that these nodal-dependent asymmetries of the epithalamus are not conserved in amniotes like mammals. We know that the mammalian brain, even though it's overall symmetrical, it's highly asymmetrical when it comes to function. And you all know that speech is, the centers for speech are largely localized on the left side, and so is the handedness is also an asymmetric function, since 90% of the population is right-handed, and there are a number of other functions which are lateralized in the brain. What is very striking is that we know essentially nothing about the control of these asymmetries in the brain and how this very compartmentalized functional organization of the brain is generated during development. So then we went back to the mouse and wondered whether this system that controls the asymmetry of the somites, this Re-dependent retinoic acid signaling pathway, could be important for controlling the symmetry and the asymmetry in the mammalian brain. And so what I showed you earlier but did not insist is that if you look at these Re embryos crossed into the RALAXZ reporter, you see that there is normally strong expression of RALAXZ in the forebrain, which can also be seen here in the cortex at E12. But this is completely gone when you cross to the open mind background, which suggests that, yes, the Re-dependent retinoic acid pathway is active during forebrain development. So, and then we became interested in that and realized that there is a very striking similarity in the way the two systems, namely the somatogenesis and the motor cortex patterning, are established. And both take advantage of a gradient of FGF8, which is antagonized by retinoic acid signaling. So this is better described in the case of the somites, but there are also a number of arguments that suggest this in the brain. And so the way the different areas of the cortex are specified, and we heard about from Alessandra, is largely dependent on the anterior signaling centers that produces FGF8, and that is going to be important for the specification of the motor and sensory areas. So we were quite struck by this analogy, and what Gonzalo did is to start to look at potential asymmetries in the brain of the mouse Re mutants. And so there he couldn't go to the homozygotes because they die at E9, E10, but then he moved to heterozygotes, and these heterozygotes are essentially normal. So they've been phenotyped on the behavior platform in Strasbourg, and they don't show any difference with the controls. But if you look carefully and if you measure the size of the motor cortex based on drawing, this is a hybridization with LMO4, which marks the motor cortex here in this area, and you just measure the distance between the boundary of the motor cortex and the tip here, what you see is that in the heterozygotes, the open mind heterozygotes, the motor cortex is slightly enlarged on the right side. So there is a slight enlargement, about 10–15%, which is seen. It's very subtle, but it's definitely statistically significant. And so this was quite, again, supporting this idea of there is a similarity in the patterning system between somites and brain, and so we decided to go one step further. And there are very few hints of asymmetry, very few ways to look at functional asymmetries in the mouse brain. And so one of the easiest ones was to look at handedness. And so it's known that most of the mouse strains show about 50% dextral mice and 50% sinistral at the population level. And so we thought that we should look at the handedness of the embryos that are mutant for Re. And this can be done using a text which is called the MORAG, where you essentially give a pellet of food to a mouse, and you watch. This is a right-handed mouse. And so you do this many times for several days, and then you come up with this sort of graphs, where here what you see is a comparison between females. So there are some females and males, wild type and het populations. And what you immediately see is that in the wild type, there's about 50-50. It's about more 40-60 rather. But there is a very striking departure from this distribution in the heterozygote animals, with almost 80% of the populations that become right-handed. Which means that and this is very much like the human situation, by the way. So this is very striking because these mouse are essentially normal, as I told you. And so far, the defect that we see is this slight shift this slight shift of the motor cortex, which would be consistent with a slight increase of the FGF gradient on this side, which is exactly what we see in the somites. And then we teamed up with Alexandra Petier and Stéphane Le Hérissi here at the Institut du Cerveau and La Moelle in Paris, and looked by MRI at the cortex of this of a set of mice which have been phenotyped for the PO usage. So these are left-handed mice, and what we are looking at are for asymmetries, for regions that are asymmetrical between the two sides, which are enlarged, in fact, on one side. So here you see that the cortex is essentially symmetrical in the left-handed animals. In the right-handed animals, you see that there is a slight enlargement of the cortex in the region of the motor cortex, which is on the right side. So again, fitting with this increase in the gradient. And in the riri heads, you see that there is an enlargement which is even stronger, but which is in the same region. And so, and these are the projections. So you see these are the left pod animal. These are the right pod animal, slight enlargement in the right motor at the region between the junction between the motor and the sensory cortex, and same region for the riri animals, which somehow suggests that losing one copy of the riri genes transform these animals into right-handed animals. So the conclusions here is that the same riri-dependent retinoic acid pathway, and I would argue that this is a non-canonical retinoic acid pathway, controls the symmetry of the FGF gradient, the bilateral symmetry of the gradient, both in somites and in brain, and probably in other organs as well. How this works in details, we don't understand yet very well. And also, I want to reemphasize the fact that removing one single dose of this gene can completely alter the behavior of the population, not the individuals, the population which become essentially right-handed. And I think this might definitely be relevant for human populations and the localization of higher functions like speech. And so I just want to stop here. And so I told you about work from two very talented graduate students, Alexis Hubo, who's now a postdoc in Novartis, and Margarita Diaz-Quadros, who's still in the lab. So Gonzalo was working with me in France, Mike Rebaglietti also. And so we've benefited from great help from Dan Wagner for the single-cell sequencing, Jérôme and Ziad for the DSCRs, Madhavan for the physics, and Alexandra and Stéphane and Hamid for the behavior. And I'll stop here. Thank you.